So, for Chapter 9, we are going to be discussing cell communication. Cells have to be able to talk to each other. Okay, because think about it. As you sit there, you have trillions upon trillions upon trillions of cells all working together in some form or fashion to form tissues. Tissues have to work together for organs. Organs have to work together for organ systems. And some organ systems have to control other systems. So in our discussion of signaling or talking, we're going to be looking at two types of signals, two types of ways of cells talking to each other. First, the talking between cells, intercellular. Intercellular is one cell talking to another cell that is not in close proximity. May not even be in the same tissue, the same organ. Could be cells talking from one part of the body to cells in all parts of the body. And then there's going to be the intracellular signals are the signals that take place within the cell. Okay, a series of enzymes and a series of proteins and little cellular molecules that move around throughout the cell, transferring the signal from one part of the cell to another. Now, when it comes to the intercellular, the signals that are going to be released are known as ligands. Okay? This is an umbrella term. These ligands could be anything from hormones to proteins to polypeptides, polypeptide fragments, or just simple molecules. They're going to be released into the surrounding fluid. That's the extracellular fluid around the cell. They could be carried off by the lymphatic system, or these ligands could then get into the bloodstream and then be carried throughout the body. These ligands are going to interact with specific molecules on target cells. These molecules are going to be lipopolysaccharides, or they could be not lipopolysaccharides, sorry, that's bacteria. It could be different glycoproteins, glycolipids on the cell surface. So when you look at the different types of signaling, whether inter or intra signaling, autocrine, cell releases ligands, cell releases signal molecules. Some of those signal molecules act on the cell itself. It's a means of upregulating or activating its own abilities. Signaling through gap junctions. Remember, gap junctions are going to be basically little windows between adjoining cells so that things could be sent back and forth. Signals are going to be one of those things. Paracrine, ligands or some signal molecules released into the extracellular fluid, and that ligand acts on cells within that same tissue, within that same organ. And then endocrine. Ligands, signal molecules, are going to be released and then carried away by the bloodstream. So a gland at the base of your brain can be affecting how different organs in your abdomen function. Your pancreas can be releasing signal molecules that are then going to be modulating the metabolism of all the cells in your body. So when you look here at this list here, this is pretty much what I was just talking about. Paracrine, released, and then, initiates signaling on the cell itself. They also add in synaptic signaling here, which we'll talk about on the next slide, where one nerve releases signal molecules that are then going to be acting on another nerve that is not quite but almost touching. So that signal from one impulse signal can go from one nerve cell to the next. Endocrine, think hormones. Hormones are released by the ovaries, the testes, thymus. Get out and then go everywhere. Autocrine, which I just screwed up a minute ago and was talking paracrine and said autocrine. Autocrine, release, activates self. And then through the signaling gap. When you look at the neurotransmitter, the signal that we were talking about is traveling in this direction. So this impulse has to somehow get across from here over to here. Well, it's not an actual electrical impulse. It's a depolarization across the myelin sheaths and all this stuff. So what happens is the impulse hits the end. And what's going to happen at the presynaptic cell is going to release neurotransmitters, small little signal molecules that are going to diffuse across this gap. The gap is going to be barely measurable. So it's going to be a quick diffusion. The signal molecules are then going to interact with a specific receptor. Specific receptor that recognizes the specific shape of that signal molecule. Once that happens, depolarization, postsynaptic neuron picks up the signal. Now, to keep all of these neurotransmitters from continuously binding and sending signals and stuff when there shouldn't be, there's going to be an enzyme that immediately also goes through and starts breaking down the neurotransmitters. So the neurotransmitters, as soon as they're released, are getting destroyed by this enzyme. So they're going to dump a lot more than's necessary so that some of it gets across. Now, not all the receptors are going to be on the surface. Later in this chapter, the ones that we'll be discussing are going to be plasma membrane associated receptors. Keep in mind, though, there are receptors that are going to be floating around in the cytoplasm, because some of the signal molecules, like hormones, testosterone, estrogen, which are highly hydrophobic, can pass through the membrane. They can pass through the membrane because, remember, when it comes to the plasma membrane or inner membranes, hydrophobic molecules can pass readily through. So they pass through, so their receptors are going to be floating around in the cytoplasm. Signal molecule binds to it, and then everything, the molecule and the receptor, will then go to the nucleus. They'll bind to the DNA and turn on certain genes.